apples? Tacos?
Like this big.
Everybody, so you know we're about 65% through the semester, right? We're almost done. And so now it's going to appear that every week we have some kind of exam. And that's because it's true. Like this always happens that way. And not only that, but if you're taking 4 other classes, it's going to feel that way the same from all those other classes also.
Yeah.
Including math. Yeah. Good. So Dr. John is going to open the lab tomorrow from 12 to 3. I cannot be here. I have a doctor's appointment. But he's going to open the lab from 12 to 3. I will probably open it, if I can, the next Friday, although I'm supposed to be interviewing people for faculty as adjuncts on that day, but we'll see what happens. Okay? I'm going to tell you right now that this practical is the hardest one of the entire semester. There's no reason for me to sugarcoat it. It is just hard, right? So you need to be ready for it. It's all diagnostic, and you're going to have to know the names of the organisms as they relate to the diagnostic test. The stuff that is on the board is the only things I will ask about organisms. And in a minute, you're going to fill those out because that is your extra credit. So if you participate in putting a letter up there, I'm watching. I will take a picture of the group when they're up there. And if you add something to there, then you're going to get 3 points. The other 2 points on this one practical only. If you study in a group, you can get 2 more, or if you go to an SI session. Okay? And Haley's going to have SI sessions online. I think you can show up if you have time. Okay? Good? So there will be no, because you know each other's names now, I can't really ask about people's names anymore because you all know each other too well. Okay, so you'll need to know the test, the setups, the substrates, the reagents, the pH indicators, the presence-absence indicators, results, pH of the results, end products, enzymes, and how to evaluate and analyze the test and what they mean. Okay? For these tests, McConkey, EMB, Hactuin, oxidase, catalase, phenylalanine deaminase, and the decarboxylase test. Remember, those are the things that we actually did in Unit 2, but we moved them to Unit 3 so that they made more sense because they're enteric identification test. The blood auger and the A&P sensitivity, the PEA, bioestin and a 6.5 salt tolerance, mannitol salt. Novobiocin, I haven't talked about it. It will not be on this practical. I will not ask about it. But it's coming. You'll get to see what it means in a little bit.
Wait, which one?
Novobiocin.
Okay.
This one right here.
Gotcha.
Hey everybody, this is Guy. Say hello. What's up with
Hi,
you? Have a good one. So, you'll need to understand the Staffy tech, UV sanitation, and then antimicrobial sensitivity. So that's also known as Kirby valve. Okay? There'll be 50 questions, just like normal. It'll be, it's a time test. You're not going back at the end, so you'll need to answer it as we go. Okay?
So we won't be able to ask questions?
You can ask questions, but during the practical. You can't, at the very end, you can't say, Toby, can we go back to number 9? No. Right then and there. Okay? So let's do it. Let's actually talk about the review. So identify the type of hemolysis, beta. So if you don't know that that's clear, you can raise your hand and you can say, Pro-V, what color of zone am I looking at? That's a fair clarification. But you can't ask things like, Pro-V, can you confirm that that is beta hemolysis? I'll give you the look, and then I'll change the question, and I'll forgive you but after 3 times of doing that, I might get grumpy. Okay? And what type of media is this? Blood agar. Good. What percent of blood is in blood agar? Let's go. So here, we're looking at what type of hemolysis? Alpha. It's green. Okay? So now we get a little bit. So here I've got a gram-positive coccus. It's that has this type of hemolysis. It's clear zone around the colony, and it is catalase negative. So that immediately puts it into a strep or enterococcus. And then it's, what is on the A disc? What is on the A disc? Bacitracin. It's showing sensitivity to bacitracin. What is this area around the disc called?
201.
Huh? Not 201, girl. It's ZO. It's ZOI. ZOI stands for zone of inhibition. Okay? So therefore, it's sensitive to the A disc. So if you got a gram-positive coccus that's beta hemolytic, it is sensitive to the A disc. What is the genus of the species? Not rhetorical, I'm expecting an answer. Streptococcus pyogenes. And what group does that belong to, Lisette? Group A. How do you know that?
Because I have my matrix.
Or because there's an A on the A disc. Are you with me? Okay, so I want you right now to show everybody the matrix. How many people have located the matrix?
It's really, really helpful.
It is very, very helpful. That is, I did that for you all. All the answers you need are there. It is. Anna? All right, let's go on. So now what type of hemolysis am I looking at? Alpha. It's a green zone around the colony. So listen, if you can't tell that that's green, you need to say, Pro V, can you tell me what color the zone is? All right? So I've got a gram-positive coccus. It's catalase negative. It is sensitive to the P disc. What is on the P disc? Optogen. What does the P stand for? No. Good guess, but no. Huh? Pneumonia. We call it the P disc because this is a test that is specific to identify Streptococcus pneumoniae. Okay? So if you know that it is a gram-positive coccus, it's catalase negative, it is alpha hemolytic, and it's sensitive to the P disc, that's enough for you to say that it is which genus and species? Streptococcus pneumonia. What group does that belong to?
P.
Huh? No. What group does it belong to? Itati. No.
B.
No.
C.
Okay, no. There's no group. It is not groupable because it has a big old capsule, and the capsule hides the sugars from the actual test. Remember, what was the name of the scientist who came up with how to identify the streptococci based on something? What was the name of the scientist? Sugars in the cell wall. What was her name? Dr. Sherwood? No. Dr. Rebecca Lansfield. And if you're looking at your matrix from your unknown, you'll see that there is the Lansfield grouping. Okay? I guess Harlow got mad at me. Okay, so is this particular organism sensitive or resistant to the P disc?
Resistant.
Okay, we're looking at the oxidase test. What is the reagent for the oxidase test? Oxidase reagent. And if it's purple within 15 seconds, is it positive or negative?
Positive.
Positive. And does that mean that it is a non-enteric or enteric?
It's not.
Good. Does it mean that it is an aerobe or a facultatively anaerobic organism? Aerobe. Because the enterics are negative for oxidase always, They're facultatively anaerobic, right? Okay? I will not ask this question because that's Pseudomonas aeruginosa, and I've never talked about it in this way. Mostly because it's too easy as an unknown that I don't give it out. If you have a blue-green pigment being produced, it's Pseudomonas. So I just don't get it. Okay? Any questions? All right, this is mannitol salt agar. If you know the name of the media, mannitol salt agar, you know a lot about it. Okay? So what's the pH indicator in mannitol salt agar? Phenol red. What is the primary substrate? Mannitol. And 7.5% sodium chloride is added to this medium for what purpose? Inhibit of everything that's not a halophile. So the staph are halophilic. So if it grows on this plate, it belongs to the Staphylococcus group, and that's Staphylococcus, Pneumococcal or Bruchura. It's those guys. Okay? What is the result of this particular organism? Right here. What's the result?
Positive.
No, you can't say positive. It's mannitol fermentor. And that is for the pathogenic organism. And what organism is the major pathogen in this group? Staphylococcus. PEA, phenol ethanol agar, what's its purpose? Inhibits gram negative. Does it kill them? Just slows them down. And it's been doing a pretty good job. This morning I was surprised that even 9 days later, it's still inhibiting the gram negative. And so I was pretty impressed with that. Okay? PEA stands for what? Phenol ethanol agar. And what is this? What type of medium is this? Selective. It's selective. Okay. Any questions? All right. This is EMB, eosin methylene blue. Again, if you know the name of the media, you know a lot about the medium, right? What's the primary pH indicator in EMB? Eosin. If you say eosin, why that's absolutely, but I'm okay if you just say eosin. Okay? Methylene blue and bile salts are added to this medium for what purpose? Inhibit of? No, not grandpa's. Yeah, grandpa's, I'm sorry. You're right. Grandpa's. Grandpa. What's the principal substrate? Lactose. If the organism produces a metallic green sheen on this medium, is it a fermenter, or is it a lactose fermenter or a non-lactose fermenter? Fermenter. And what specific organism does this? E. coli. And this medium is designed to do what? Test water for fecal contamination, of which E. coli is the marker. Okay?
On the matrix,
Yeah.
it says
Oh, that was just a note that was put in there by a student who I gave permission to change it. Yeah, you can do that. Okay? It does say lactose fermenter though. All right. Anybody else have questions? Okay, this is McConkey agar. What is the application for McConkey agar? It tests all clinical specimens. A or B, which one is a lactose fermenter? B. So the non-lactose fermenter will be A, be colorless. Okay. Crystal violet and bile salts are added to this medium for what purpose? What is the pH indicator? No, not phenol red. Thank you, Nikki, for trying, but no.
Neutral.
Neutral red. Neutral red. Okay? This is the catalase test. What is the substrate in the catalase test?
Hydrogen.
Hydrogen peroxide. What is the reagent in the catalase test? Hydrogen peroxide. A or B, which one represents streptococcus? B. B. Streptococci and enterococci are negative. Staphylococci, micrococci, and Klebsiella are positive. Which medium should you never perform a catalase test from? Blood agar. Because blood agar has its own catalase activity, it'll give you a false positive every single time. True or false? You should use a metal loop when doing the catalase test. Right, it has to be something non-conductive. Metal will react and give you a false positive. Okay? This is the Staffy Tech. I'm going to tell you the answer to a question that's going to be on the practical, and you're going to give me something else that's crazy, because you always do. What type of test is this? Antigen antibody. It's an antigen antibody test, right? This is the one that we have clumping of the cells by the immunoglobulin, right? So these little Y things are the immunoglobulin. They're not binding, and here they're binding, and you see what it looks like when it gets bound up. It gets what we call agglutinated, okay? This is the same thing y'all did when you do blood typing. Okay? Okay, what is this called? Kirby-Bauer or antimicrobial sensitivity or susceptibility testing. Those are all fair answers. Okay? What is this growth here called?
Which one?
This growth right here, along. The other more fancy word for it is confluent growth. Okay, mean the same thing. Okay? So, what is the standard of turbidity used to make sure that we have the proper inoculum? It's not rhetorical, I'm expecting an answer.
What was the question?
What is the standard of turbidity to make sure we're using the correct population?
1/3.
A 0.5 or a 1 what?
No more than.
I don't really care about the grades, I mostly care about the name. And the name is what? These things actually exist. This is a half McFarland. Okay? It's a McFarland. McFarland. Okay? What is the clearing around the disc called? Don't call it a 201. A ZOI, that's correct. So, based on, because if you were going to measure these things, what units would you measure them in? Millimeters. MM is good. So if we're not going to measure them, so based on just observation, which lettering represents a disc that shows complete resistance by the organism? No, resistance. Be sure you understand the question. SSS. Which one shows complete susceptibility? Now P. And what is this stuff called right here? That's growing in the actual zone.
We have the colony within the colony.
What is it called? Yes.
Breakthrough.
Breakthrough is correct. And should you, true or false, this is a good choice for an antibiotic to fight the infection of a person who has this organism. True or false?
You wouldn't use it for anything else.
6, which is a third generation cephalosporin. This particular antibiotic is a good choice to treat somebody who's got an infection by this organism. False. It's resistant. And it might not be totally resistant, but there's enough organisms that are breaking through that shows that it's not going to work in
You
this.
said if you had it on hand, that's all you guys did it, but it was.
No, I mean, I would go get something else. I would never give this. The patient's going to die. If you give him this, the patient's going to die. Okay? It's just the way it is. I'll go to some other country and get some other drug that we don't have here before I would give him this. Put me on a plane, I'll go get it.
But it'll just cause more resistance to
It won't cause more resistance, it's resistant. But no. It doesn't need to get stronger. It's as strong as it's gonna get. Okay? But I mean you just never would give this. Okay? Alright, here we go. True or false? When you perform a KB, it's okay to use a mixed culture. What does that mean, Annalisa? Is that true or false? Well, that means no to me. It doesn't. Okay, false. Yeah, because if they both are sensitive, okay, but that's unlikely. One of them is going to be resistant, and then you're not going to know which one. You're just wasting time. Okay. So this is Hector enteric agar. What is the pH indicator in Hector enteric agar? What's its application? What's it used for? To screen for pathogens in stool. And what are the two primary pathogens in stool? Poop. What's the two primary pathogens in poop? Salmonella and Shigella. We don't bring Shigella out at all here because it's too mean. What I mean by that is, to get an infection by Salmonella, you would have to come in contact with about 100,000 cells. To get an infection by Shigella, 10 is all you need. It's not a good bug. We don't bring it out. Okay? So what is the result of this organism here? It has 2 parts. What is the result? It's a non-lactose fermenter and hydrogen sulfide positive. If you leave one of those off, it's wrong. Okay? These are lactose fermenters. True or false? These are really important as pathogens in the GI tract. It's not rhetorical. I'm expecting an answer.
false.
They are normal. There is a few types of E. coli that are bad, right? But these, for the most part, are normal. So the Klebsiella, the Citrobacter, all those other things. How do you know it's normal? Because it's a lactose fermenter. Remember, I put on the board, we only worry when we have a lactose non-fermenter or a non-lactose fermenter that's hydrogen sulfide positive. If I ask what is the major pathogen on this plate, you would say what? On this plate. Huh? What is the major pathogen on this plate? Salmonella.
E. coli was the yellow-orange, that's one.
Yeah. So be sure you know be sure you know what, you know that little chart I put up for bile esculin at 6.5? Yeah, I don't know that. Okay? But specifically, the most important organism here is bioesculin positive and 6.5 salt tolerance positive. So if you have an organism from an enteric, it's an enteric culture, right? So it was taken, I don't know, from someplace. And this was actually causing, I don't know, infections, I don't know, the heart or the ears or whatever. But it's a gram-positive coccus that's either gamma or alpha hemolytic. It's catalase negative. It's bacitracin resistant, alphagen resistant, and it's bioesculin positive, 6.5 positive. What is the genus that's involved here? Enterococcus. And what group does it belong to? Group D. Group D. Correct.
It also says H or K.
That is gonna be if it's bioesculin positive but 6.5 negative. If it's bioesculin positive and 6.5 positive, then it's just gonna be enterococcus. Okay? So here is an organism that's beta hemolytic and it's represented by group B. What is the genus and the species of the organism? Streptococcus, what? Hey, go back. We talked that, we did that. Be sure you know the decarboxylase test. What is the pH indicator in the decarboxylase test? Bromocreosol purple. What is this stuff up here?
But that's the oil.
Mineral oil. Don't say oil. Mineral oil. Why is it there?
It's just that oxygen.
An anaerobic test, because this decarboxylase tests are only valid if they're anaerobic. Okay? Name one of the substrates that could be in the decarboxylase test. Arginine, good. Name another. Lysine, very good. Name the last one. Ornithine. I'm gonna ask to give me one of them. Pick one and write it. Okay? Which one is positive, yellow or purple? Yellow or purple? Purple. What's the pH of the purple tube? Acidic, basic, or neutral? Basic. What's the pH of the yellow tube? Acidic, basic, or neutral? Acidic. And then there's the phenylalanine deaminase test. It presents as a SNAT. How are you going to inoculate it? SSSD. Very good. And after 24 hours, you come back and you add a reagent. What reagent do you add? Ferric chloride. What color is positive? What color is positive? Green. Any other color is negative. Okay.